we move together from ears to the cortex, and now we have the information in the cortex. And then the question is, okay, we have this huge amount of information from the environment. We can go back to the scene that we discussed first part of the class, that busy, crowded jazz festival in the streets. That people are talking, the music is going on, and there are many kind of different sounds that are mixed together. And we are kind of recording these pieces of information, and now they are on the cortex. How we put different pieces of information together, and we separate other pieces from each other to be able to understand what is happening. So we have what we call grouping and segregation. So there are occasions that we need to mix different pieces of information together, and there are occasions that we need to just separate them from each other. How these grouping and segregation are happening. Okay. Let me kind of explain the problem for you. For example, within the same jazz festival, you have 3 artists on the stage that are playing, and you can see that they are making different kind of sources of sounds. Okay, so we have different sources of sounds on the stage that are making the sounds, and then what you perceive is a mixture of these 3 sources together as a sound. And then what you are supposed to do is just taking this, mixing some of these frequencies to number 1 stream, number 2 stream, and number 3 stream. So you need to do both combining different pieces together, grouping them together, because we discussed there are just frequencies here. So some of these frequencies are supposed to go to make the sound, the first stream. Some of these frequencies are going to make the second stream, and some of the others are going to make the third stream. And the question is how our brain is able to do that. And in reality, I should tell you that nobody knows clearly how this is happening inside the brain. So we just have kind of pieces of information that they're kind of about the potential mechanism of how these kind of complex auditory processing is happening inside the brain. But in reality, we really clearly don't know what is really happening in terms of how these pieces are being, in such a beautiful sense, combined to each other and then separated from each other as well. Okay, there are pieces of information I'm going to kind of share with you and discuss about, and just give you a sense how we make pieces of information, how we try to answer to this question with experimental paradigms. And if you ask me at the end of this class, what are the qualifications I'm expecting you to be able to have? Probably one of the most important kind of qualifications I'm expecting you to have is being able to read a human subject cognitive neuroscience study and understand what is happening there. Not only the beautiful pictures, but also to understand what are the things that are being presented in the paper. In the textbook, in the kind of Fundamentals of Cognitive Neuroscience textbook, involved the pages, we have a figure. Probably I can find the figure in page. It is in page 164. Okay, this is the figure that is present in the book. It says control, name, segregation. It's just a little bit complex to understand. First of all, you need to go back and see the actual paper that you have here. If I go to the actual paper, even from the title of the paper, you can see that this paper is about a comparison between a group of people with Alzheimer and a group of control subjects. So that's the reason that we have control here, because they had another Alzheimer patient group as well. So that makes sense to have both Alzheimer and control subjects, okay? And then the question was about auditory scene analysis, how the auditory scene analysis is happening. The journal is Neuroimage Clinical. I'm not sure if there is anybody who knows the impact factor of Neuroimage Clinical. You can double check that and just type it in the chat space. What is the impact factor of this journal? Which is a really good journal in the field, which is a kind of a branch from Neuroimage, which is a kind of the parent journal, and then Neuroimage is Clinical is a division from Neuroimage. And it has been published in 2015, so it is pretty much a new publication. And even in a new publication, the question of auditory scene analysis is a valid question. So it's important for us to know that even, this is something like an active question in the field that people are still exploring about how to kind of test that. The paper, the basically textbook is reporting that with these areas that are in cyan or blue, they are related to, let's say, what we call grouping and understanding your name in a complex, busy, noisy environment, okay? And then these magenta or purplish kind of areas are related to segregation process. And how segregation is being separated from grouping in experimental design is an interesting question, okay? I highly recommend you to see, those who are interested in these areas, you can basically see the paper. Here is the kind of the design that they had. They were presenting name of people in two conditions. One is an interleave, I is interleave, so name interleave, okay? It means that this one is noise. This is noise, and then they just have the name without any noise. So they call that interleaved. So noise, name, noise, name. So the name is not confounded or distracted by the noise. Here, this one is superimposed. So name is superimposed to the noise. So here, people need to be able to do segregation. So there is no need for segregation here. There is no need for segregation here, okay? And then they have reverse name, which basically spelling the name from the backward way. And then it is totally nonsense, okay? And then they have that with superimpose and also interleave, okay? So they had basically 4 different conditions, okay? Based on these 4 different conditions or types of trials that they have, they claim that they are able to differentiate grouping from segregation processing. How? They believe that when we have grouping, so we need to basically put the information together and match that with a template in the brain. And the name identification is a good example of grouping information. If we are not able to group pieces of information together, we cannot identify name. So by identifying name, we mean that we have been able to group things together. And then they believe that if we kind of do the segregation, is when we are doing the name identification. We do name identification when we are presenting name, whether it is superimposed or it is interleaved. In both conditions, we have name identification. And these are basically the control conditions because we do not have name. Here we have name. Here we do not have name, okay? So if we contrast them, that is going to be name identification. And that is technically these cyan pieces, which is bilateral, and it's related to the primary auditory cortex, and it is here, okay? But where I am doing the segregation. Basically, I'm doing segregation here when something is superimposed. I need to do segregation here. And where I do not need to do segregation. Here, there is no segregation because they are technically interleaved. There is no need for segregation. Okay? Do you agree with me for that? And they made that contrast. I prefer to have this one, which has already, when we have superimposed, we have segregation. There is no segregation here. I prefer to have this one minus this one. What they have here is this one minus this one, which is, I can imagine why they have done that. I'm not going to discuss about that, but they have done that because of being able to report positive results, a positive kind of contrast, not negative contrast. There are some technical issues for that. But the reality is they are basically kind of making the contrast. So in this design, which is a good sample of what we call factorial design, this is a factorial design. They try to kind of, first of all, see these two main effects. And then they claim that the interaction between these two effects is what we call the cocktail party effect. And the cocktail party effect is exactly what we have been discussing about before here, that we do both, we are doing segregation while we are doing the combination. So while we are basically combining things together and segregating them together, that is going to be interaction between these two effects. And this is the interaction. We call that cocktail party effect. And that is going to be happening in an area out of the auditory cortex, in an area which is called, I will tell you what is the name of this area, but this is an area here. As you can see here, this is out of the temporal cortex. It is in the parietal cortex. Okay? Does it seem complex? It seems, it is a little bit complex. And let me just kind of bring you the actual paper. So this is the actual paper. A group of people in UCL have done that with 17 subjects in the control and 13 Alzheimer patients. What they have, they reported that the results in healthy control, Alzheimer patient, and patient over control, and they showed that technically healthy control and Alzheimer patients are the same for all the effects that they had. The only thing that is different between Alzheimer patient and control patient is about the cocktail party effect, which is weaker in Alzheimer patients. I can discuss about that later. But what I'm really interested for you to be able to do is just being able to read this table. So first of all, you need to talk about different contrasts based on what I showed you before. This one is sound versus silence. So in sound versus silence, what they are doing, basically combining everything that they have been kind of showing, to kind of presenting to the subjects, and then comparing to the silence. So just basic sound versus silence. In sound versus silence, these are the activations that they have. HG, that you know what HG means. They have mid STG, you know STG means superior temporal gyrus. And then STG and STS. So these are the pieces that you basically know those pieces. And technically, it is the same for Alzheimer patient and healthy control. And then they have a name identification, segregation, and then at the end, they have the cocktail effect party, which is located in SMG. Okay? And what is SMG? It is supramarginal gyrus. How can I find supramarginal gyrus? I just open a page. I just Google for supramarginal gyrus. I just bring the images here. I can just open one of these images. Let me just open this one. So here, let me open this one. Just, oh, it's not the, okay, so here you can see this one is called supramarginal gyrus, this area. And then there is another gyrus, which is called angular gyrus, which is very similar to the gyrus that we discussed about with the name of lingual gyrus. Do you remember where is lingual gyrus? Those who have done their homeworks from last session and explored the brainotome atlas, they can tell me where is lingual gyrus. This one is angular gyrus. Where is lingual gyrus? You can type it in the chat space. Okay, to see how many people they know about that. They remember where it is. Those who are fast, they can just go and see that in the brainotome atlas. But yeah, probably if I'm talking about the kind of last session homework, you know that the last session homework was about occipital cortex. So probably we know that lingual cortex is in the kind of occipital cortex. But I mean, those who are interested, they can go back and see what they have in the kind of medial occipital cortex compared to a lateral occipital cortex. And this one is in the medial, exactly. It is in the medial occipital cortex, yes. So it is in the medial side of the occipital cortex, not on the lateral side of occipital cortex. I'm not going to just make you confused, but this one is angular gyrus, this one is supramarginal gyrus, but this is what we could see here in terms of, this is the activation that we have here. Does it seem complex? Yes, it is, to some extent complex in terms of extracting the information. But in reality, sometimes just having one single figure and just being able to use that one single figure to understand about what do we have in textbook sometimes is a little bit complex. And I highly recommend you to see the citations and references that are provided here. But just going back to the question, so we are trying, so people, scientists across the world are trying to understand how this complex phenomenon is happening in terms of what we call the cocktail party phenomenon. How we process the information in a cocktail party when different people are speaking, and we just merge different pieces of information together to decompose, to compose again, or recompose again somebody's voice and understand what is happening, compared to kind of segregating that with the voice outside. So there are still complexities for that, and there are pieces of information that these kind of pieces of information are happening in different parts of the auditory cortex. I'm going to give you more details about that in the next slides. This is kind of the last slide for this part, but it is giving lots of kind of information. I'm just going to give you a sense about what is happening in this kind of auditory processing, and just stop here in terms of the anatomy, and goes for, then we can go for other levels of processing. So what we have here, so this one is primary auditory cortex. And I think that we have learned that very well. So this one is the Heschel gyrus, which is the primary auditory cortex. And then we have secondary auditory cortex. And then going out of this primary and secondary auditory cortex, then the other things around are not only auditory. They have different other information that are combined together. And then we have two lines. One line which is going from this side to the ventral side. This called is ventral pathway. And another line which is going to the back and to the dorsal part. So we have a dorsal pathway. It is similar to the ventral and dorsal pathway for the visual system, but this is an auditory system. Here we have areas related to language production. Language production. And these are areas regarding to language understanding. And these two kind of systems, or these two pathways, which are kind of circling in two different directions, are processing information when we try to understand what is happening around us, to talk, or understand what we have been talking about, or understanding about what people are talking about, and start to process that. This comes from a Nature Neuroscience paper that is talking about the forward mapping and backward mapping in this system. As you can see here, the area 41 was the core primary auditory cortex. Area 42 was the secondary auditory cortex. And then area 22 was the area that was involved in kind of combined multimodal processing. And then we have IPL, inferior parietal lobule. And then we have other pieces. And this one is primary motor cortex. This one is primary motor cortex. And this one is premotor cortex. So do we have anybody who knows the Brodmann area of this area, this part? The Brodmann area number what? Okay, anybody could say that in the chat space. And then we have area 44 and 45. And we discussed about these areas before, if you remember, when we discussed about inferior frontal cortex or inferior frontal gyrus. And there are some other technical details here, like kind of here we have superior temporal sulcus here, which is also another important area here in this kind of map. But that is basically just a brief overview about the segregation and matching of different pieces of information in the visual system to be able to understand what is happening in this auditory processing. And as I told you, there are definitely lots of complexity in terms of how we make a representational space, a conceptual representational space about auditory kind of environment that we have, which is not going to be really easy to do, but as complex, as kind of many challenges that we have for the visual system, we have really interesting challenges for the auditory system. And sometimes we are more focused as something like cognitive neuroscientists to the visual system, but we have the same level of complexities in the auditory processing as well, which is sometimes being neglected to some extent. But we have a really complex auditory processing system as well. And then we have Marina. Marina, as you are a psychiatrist and you have done all the kind of the medical school exams and the other things, and you have learned all these kind of pathways before. For you that you already have that kind of medical background, how much does it seem complex to you?
Yeah, it was like quite a long time ago when I was in medical school, and actually it's very helpful that we have like Latin term, and the most of information is kind of easy for me to understand, but there are still so I actually think that for me it's quite hard to understand, and because I'm not really familiar with like neuroscience papers. I never read them, so it's basically like physiology and classes in anatomy or physiology, and it's quite different. So yeah, the anatomy terms is not hard for me, but to understand the mechanism of how it's working and to be able to ask the question like why it's so important to have like descending pathways. Like for me, I've never asked myself these questions because in medical school you just need to have a good memory and like reproduce this information. But like you ask some very interesting questions like why it's working this way, and I become like more conscious about like exactly like why our brain work like this. And it's actually like for me it's quite I just wondering like how everything like it's very complex, but it's very smart system. And yeah, but it still for me it takes time to understand it. So yeah,
That's good. That's good. Now, I mean, all other kind of course participants, that is making things a little bit easier for them because they understand that even if you have a strong medical background, there are still complexities in terms of understanding these concepts and all these kind of information flows and pathways and how these pieces of information are working together and how we can understand a kind of a scientific paper with these, let's say, methodologies and kind of anatomical descriptions and coordinates and contrast and experimental designs and scientific questions. So for those who feel that it's too complex, welcome to the club. So it is the same problem around the world. So it just it is not something that you are alone in this. We are all together in this. So it is just the process. Kiana, go ahead. Do you have any question?
Yeah, about the paper you showed in the Golden 2015 Neuroimaging Clinical. You talked about adding and subtracting different bold signals, probably. The problem I have is how do we assume that this is a purely additive sense of processing? I mean, is it multiplication? Is it logarithmic? Is it linear, non-linear? I'm not sure. Do we have evidence for simple additive properties of this difference?
Initial assumption that we probably have a linear effect. I mean, if we go beyond the linear effect, things would get very complex. So the initial assumption is that there is a linear effect. But it's not a simple linear effect. So if it was a simple linear effect, there was not any sort of interaction. Here we can have interactions. So it is not just something like a simple addition. So we know that when we add something to something kind of else, based on the context, that could make a different level of kind of contribution. So kind of adding A to B in the M context compared to adding A to B in the N context would have different effects. And that's the reason that we have interaction effect. And that is something that make the factorial design studies, as we discussed in I think it was in chapter 3, right? So we have been discussing about the factorial design. There are complexities for that, as we have been discussing that we have the two main effects and we have an interaction effect. And that makes things a little bit complex. But whatever we are assuming is just basically a linear effect. We are not talking about, if we go to that level of going out of the linearity of these effects, things would get really complex. And sometimes they are not really linear. So kind of linearity is sometimes a basic assumption.